Life Sciences, Week 7, Video Lecture 13, Energy Production, Part 1. Okay, so we just looked at, in class, we just looked at the cell membrane and all of the many, many things that the cell membrane does. And so one thing we might ask ourselves is how does the cell membrane how does it make those movements? How does it do those things that it needs to do when those things require energy? So, for instance, we had looked at passive processes, ways for things to get across the cell membrane that just happen without any added energy. But then we also looked at active processes, of which there are several, and those were ways that things get across the cell membrane when energy is required, like if something is going against the concentration gradient or something like that. So where does that energy come from? Well, of course, it comes from ATP. And where does the ATP come from? Well, the cell makes it. So making ATP is one of the main things, well, one key thing anyway, that cells do. And so that's what we're going to start to take a look at here in these next two lectures. So our objectives for this lecture are: describe the role of ATP in cells, define the terms cellular respiration, aerobic respiration, anaerobic respiration, oxidation, reduction, redox reaction, and glycolysis. We'll list the three parts of cellular respiration, outline the basic steps, reactants, and resulting products of glycolysis, and state where and in what condition glycolysis occurs, and indicate where in the cell the Krebs cycle takes place and the basic reactants and products of the Krebs cycle. So that seems like a lot, doesn't it? We'll be fine. All right. So energy production. So there's an intro in the notes about the very many things that the cells do that we are oblivious to in our conscious life, in our conscious mind. And so taking a peek at a couple of these things, like making ATP, and a couple of lectures from now we'll be looking at protein synthesis, that gives us a little bit of an idea of the massive amount of activity that happens in cells all the time that we have no conscious awareness of those things happening. So making energy is a really important part of what cells do, because if they didn't make ATP, they wouldn't be able to do all of the other things that they do. So we know that ATP is the energy currency of the body. We've heard that term before. So like this slide says, it's like money that can be spent throughout the cell. So when we take in nutrients, we need to break those nutrients down, the macronutrients anyway. We need to break those nutrients down. Like, they all contain energy. Carbohydrates contain energy, lipids contain energy, proteins contain energy stored in their bonds. And then we need to break those bonds to release that energy. But before we can use it, we need to harvest that energy and put it into an ATP molecule. So ATP is the currency that the body uses. It's the, and I mean currency like money, not currency like energy, which is, I guess, that's sort of a confusing term. But anyway, that is, so we have to change it into ATP before it's something we can use. And then ATP is the form of energy that all of our cells can use. And so what ATP is doing is taking chemical energy and converting that to kinetic energy. So that's the only molecule in the body that we have that does that transition. And that's part of what makes ATP a very important molecule. So since all cells need ATP, all cells can produce ATP. And so in our process of looking at this process of ATP production, we're going to look at glucose as the fuel source. Now, eventually we'll look at other things as fuel sources as well, but glucose is kind of our, it's sort of a common starting point. We'll see a couple of reasons why for that. Well, I'll tell you a couple of reasons. One is that all cells can use glucose as a fuel source, so it's a good thing to study to see how cells use glucose to make ATP. And likely it was the first molecule that was studied in the process of trying to unravel this really complex process of how cells make ATP. So this slide gives us an idea, a kind of a visual of what's going on with ATP production. We're taking in chemical energy from the foods that we eat. We're converting that to ATP. In that process, and then the ATP is being used for our body's metabolism. So any chemical reaction that needs to occur, any movement that needs to occur, that is going to happen by using that energy in ATP. Byproducts from this whole process, we have chemical waste products, which are carbon dioxide and water. Water, I don't like to call it a waste product as much as a byproduct, because we are going to use that water for other things. And then we have heat that is released too from that process of making ATP, and then heat also that's released from the process of whatever other reaction that ATP is going to fuel. So basically, the process of making ATP is called cellular respiration, and cellular respiration is exactly why our cells need us to eat and need us to breathe. So cellular respiration is ATP production. It can be done with or without oxygen, but to make enough to really get things done in our body, we need to do it with oxygen. So we need to take in oxygen for that process of making ATP. The process of making ATP has the byproduct of carbon dioxide, so we have to exhale that carbon dioxide, because otherwise that's going to make the blood acidic. We've seen that in buffer systems. So that's why we need to breathe. We need to bring in oxygen to make ATP. We need to exhale carbon dioxide, which is a byproduct of ATP production. And we need to eat so that we have fuel, so that we have these macronutrient cells to break down the carbohydrates, the lipids, etc., to break down, to release the energy in those bonds, which is the energy that we're then harvesting to put into the ATP molecule. So cellular respiration, so respiration, sounds like breathing and does refer to breathing, but cellular respiration is what cells are doing with the oxygen that we're bringing in, and what cells are doing with oxygen is making ATP. So let's take a look at this process. So recall when we looked at photosynthesis. So in photosynthesis, plants take in energy from the sun, they take in water through their root system from the soil, and they take in carbon dioxide from the air. And they use those reactants to create their photosynthate, or their product, which is glucose, and oxygen is the byproduct. And this slide is actually only showing the oxygen byproduct, but remember our equation that we looked at here, 6 CO2, so 6 carbon dioxide molecules, plus 6 H2O, 6 water molecules, plus light energy, so that energy from the sun, that converts to C6H12O6, which is glucose or other carbohydrates, and 6 oxygen molecules. So that was our equation that we used for photosynthesis. Now, if we turn that equation around, basically if we just flip the arrow, but on the next slide I'm going to show the equation completely flipped around, not just flipping the arrow. So if we flip it around, we have the equation that shows us cellular respiration. Okay, so let's turn that equation around, and here is cellular respiration. So we start with C6H12O6, which is glucose, and also we need 6 oxygen molecules to make this happen, and then we're going to break down or catabolize that glucose and turn it into 6 carbon dioxide molecules, which are a byproduct that we'll exhale, 6 water molecules, which are a byproduct that we may exhale, we'll likely use in various ways, and ATP energy. So this slide sort of shows that ATP, it shows it as energy. It almost looks like a sun there. So it's kind of like taking that energy from the sun that the plants harnessed and put into glucose, and then we're going to break that glucose down and release that energy again and have that energy available for us. So recall that, however, when Mr. Anderson took us through all of the steps of photosynthesis, it wasn't as simple as this, right? It wasn't just, let's take carbon dioxide, let's take water, let's put a little energy in and we'll get some glucose and some oxygen. There were many steps along the way. There were 2 different cycles. There was the light reaction and there was the Calvin cycle, and there were products from one that were needed to run the other, etc. So that was what happened with photosynthesis. With cellular respiration, it's the exact same thing. It's not as simple as this equation makes it appear, but this equation is the bottom line. This is sort of factoring everything else out. What do we need to go in and what comes out as a final product, so this is a nice way to think of cellular respiration. But we're going to get a little down and dirty with it and get into some of the more specific reactions that are happening there. So cellular respiration is actually divided into three parts. The first part is called glycolysis. Glycolysis happens inside cells in the cytosol, so not inside any particular organ, organelle, just inside the cytosol of cells. That's glycolysis. It's the first step. It does not require oxygen. The second step is the Krebs cycle. The Krebs cycle only happens if there is oxygen around, and it takes place in the matrix of the mitochondria. So the products of glycolysis are going to enter the mitochondria, and inside the mitochondria, then the Krebs cycle will happen. Then the products of the Krebs cycle, still there in the mitochondria, will enter into the third part, which is the electron transport chain. The electron transport chain also takes place inside the mitochondria. And so one thing we know about cells is some cells have more mitochondria and some cells have fewer mitochondria. And the cells that have more mitochondria are going to have a greater capacity to produce ATP. And those that have fewer will have less of a capacity to produce ATP, which is fine because cells are basically going to have the number of mitochondria they need to meet their energy needs. So anyway, so eventually we'll be taking a look inside the mitochondria here as we go through all of these steps. So there are these three parts to cellular respiration. Each of these parts is made up of like 7 to 10 or so different reactions that occur. And so before we get into those reactions, and let me tell you this, I'm not going to ask you to like write out every step of the Krebs cycle if you've ever had to do that for a class before. Basically here, this is cellular appreciation day. I want us to have a good appreciation for the things that the cells do and how complex these processes are, and how any step along the way we could sort of throw a wrench into this process and stop the whole process. So those are the things I want us to kind of keep in mind and be aware of, and just sort of kind of sit back and let the steps of this process flow over you a bit, just sort of on an appreciation level, if that makes sense. Okay, so we need to define a few terms before we can go through and talk in more detail about what's happening with cellular respiration. So let's start with our term cellular respiration, even though we already defined it, but we'll do it again. It is this is a different definition, but that's okay. This is cellular respiration is the catabolism of organic molecules to make adenosine triphosphate, which is ATP. So that means it's the breakdown of organic molecules, and specifically we know it's the breakdown of amino acids, or the breakdown of glucose, or the breakdown of fatty acids in glycerol, or also we haven't talked about this. There are some waste products that are byproducts of other processes in cells that also can go into this process of making ATP, because cells are really cool that way. They're very into recycling. They don't, if there's a molecule around and they can use it somehow, they're going to use it. They're not going to just sort of let it go to waste. So anyway, but what cellular respiration is making ATP, is the catabolism or breakdown of organic molecules to release the energy in the bonds of those molecules, harness that energy and channel it into the process of making ATP, so that ATP molecule will store that energy. And then we know that ATP, when it does its job, what it does is it transfers its chemical energy, or the energy that's stored in its high energy bond, it transfers it to another molecule, and that creates movement in that other molecule. So it is this transition from chemical energy to kinetic energy, the energy of movement. That's what ATP does. All right, here's some of our terms we need to get under our belts before we can talk about what's happening here. The first is aerobic respiration. Aerobic respiration is the production of ATP using oxygen. So aerobic uses oxygen. When we do aerobic respiration, it's a far more efficient process than anaerobic respiration, which is why we do aerobic respiration. And it's also what we need mitochondria for, because that's where aerobic respiration happens. In contrast, anaerobic respiration is the production of ATP without the use of oxygen. And our cells can all do that too, but we just don't make as much ATP doing anaerobic respiration. Glycolysis is that process of breaking glucose down into pyruvic acid anaerobically. So we'll take a look at this, but when we looked at the 3 parts of cellular respiration, glycolysis is the first of those 3 parts. So glycolysis is the anaerobic respiration part of things. It is the first step to cellular respiration, and it is a required step that needs to happen before the Krebs cycle and the electron transport chain can happen. So literally what glycolysis means is glyco refers to sugar and lysis means breakdown. So glycolysis is the breakdown of sugar, or the breakdown of glucose more specifically. It occurs in the cytosol of cells and does not require oxygen, so it's anaerobic. Okay, here are a couple of new terms. Oxidation. But you've heard oxidation, right? When you cut an apple and you leave it on your counter and you come back later and it's brown, you go, oh look, my apple's been oxidized. I know that you say that. We all just say that. And the same thing with avocado or potato or whatever. You come back later, it's brown. You say, oh, it's been oxidized. So what does that mean? It means that some electrons have been lost. It means that there's a decay process that's happening just from the exposure to oxygen. So in the case of an apple or an avocado, it's the exposure to oxygen that's doing it, which is where that term oxidation came from. But as scientists discovered these crazy little things called electrons, we realized that it wasn't just straight up exposure to oxygen that was making oxidation happen. So we still use the term, which sounds like it has to do with exposure to oxygen, but now what oxidation means is the loss of electrons. And we recognize the loss of electrons as a decay process in molecules. So when a molecule loses electrons, it loses energy. We can see oxidation happening in other things, like if you leave metal outside and it rusts, over time, or like bronze or copper having a patina forming on it. Those are all oxidation processes happening. And you can think of oxidation as a very slow burning. Like if we, had a camera and we focused it on a shovel rusting over the course of a decade, and then we went back and watched it really quickly, it would look like that shovel, the metal of that shovel was burning. So it is a process of releasing energy, which is what burning, if you have a fire going, you're releasing energy from the wood that is the fuel for that fire. But it's happening really quickly, so quickly that you can feel the heat coming off of it. In an oxidation process, at least the ones that we're talking about here, it's a much slower process, but it's the same thing. So it's the loss of electrons from molecules, which means it's the loss of energy from molecules. So those molecules are breaking down and decaying, and there's energy being released or being lost in that process. Okay, that's oxidation. The sort of flip side of oxidation is reduction. So reduction is the gaining of electrons. And so when we add electrons to a molecule, we increase the energy of that molecule. So reduction can be a little bit of a weird term, because reduction sounds like something's being lost, but it's what's actually happening with reduction is it's gaining electrons. So it's gaining a negative charge, and that's why we call it a reduction. It's having a reduction in charge, but it's gaining electrons, and also then therefore it's gaining energy. So when a molecule is reduced, it has more energy than it had. It has taken on an electron, and therefore it has taken on energy. Okay, so reduction is a gaining of electrons and a gaining of energy. Now, oxidation and reduction happen together. So if one molecule is oxidized, in other words, one molecule loses an electron, another molecule is reduced. So some other molecule gains that electron. And again, remember that losing an electron means losing energy, and gaining an electron means gaining energy. So when both of those reactions occur together, we call that a redox reaction. It's an oxidation-reduction reaction that we abbreviate to redox reaction. So in a redox reaction, one molecule loses an electron, and another molecule gains that electron. So one molecule has been oxidized, another molecule has been reduced. And that is the transferring of energy from one molecule to another. So in the process of ATP production at the process of cellular respiration, there are a lot of redox reactions that occur. There's a whole lot, because essentially that's what we're doing, right? We're breaking down a molecule, and so to do that, we're releasing the energy, so we're pulling electrons out of there, transferring it to some other molecule, and eventually that's going to be transferred to the process of making ATP. Here's our last term. I think it's our last term. Yep, is phosphorylation. Phosphorylation is the addition of a phosphoryl group to a molecule. I'll often refer to it just as the addition of a phosphate to a group, but it is actually to a molecule. It is actually a whole group, a phosphoryl group, which is phosphate and oxygen, more than one oxygen atom together in a small molecule. So phosphorylation is a word that you hear in a process that occurs in the process of making ATP. So if we have ADP, adenosine diphosphate, if we phosphorylate that, in other words, we add a phosphoryl group to that ADP, we turn it into ATP. And that's what we're doing when we're making ATP, is we're doing this phosphorylation process. We're adding this phosphate group to turn it into ATP. So that's a term that we'll hear at the end of all of this. Okay, so let's start with glycolysis. So glycolysis, let's see, I'm just finding where we're at in the notes and making sure I haven't missed anything too terribly important. Okay, I think we're ready for glycolysis. So glycolysis, like I already mentioned, is the first of the three parts of cellular respiration. So again, it's glycolysis, then the Krebs cycle, then the electron transport chain. There are, 10 steps, I think, of glycolysis, and we're not going to go through every single one, but we are going to look at them. Here they are. But we're just mostly going to summarize what's happening here. So let's zoom in. So what we're starting with here on the very top is glucose. So on the top right, we see a glucose molecule. That should look familiar, that ring of carbons. That's a glucose molecule. And then right in the center on the top, we see the word glucose. So we're starting with one glucose molecule in glycolysis. So the first three steps that occur, that are numbered 1, 2, and 3 here, those first three steps actually require 2 ATP molecules. So to make ATP, we have to put some energy in to get this rolling, which of course makes sense, right? We've talked a lot about activation energy. Activation energy is energy that you put into a system to make a reaction happen. Now we already have heat in our cells, and that adds, that makes it so that we don't need as much activation energy, but we actually have to put ATP in to get this process rolling. So in these first three steps, we're going to use 2 ATP molecules, which you can see there on the left. And so what's happening in these first few steps is we're turning that original glucose molecule in, we're actually adding a phosphate group to it, and we're turning it into a different molecule. So we could follow the molecule down on the right and see that glucose gets a phosphate added to it, and another little something added to it. Okay, so there's a few steps of adding some stuff to this glucose molecule, which is kind of funny, but that's what we do. And it requires ATP to do that. The next few steps 4 and 5, what we're doing there is that we're actually splitting that glucose molecule in half. So after the first 3 steps, or maybe it's after the 4th step, we have the glucose molecule has changed into a carbohydrate molecule that is symmetrical. And we're going to take that symmetrical molecule and split it in half so that we have 2 of these same molecules, okay? And these 2 molecules are called glyceraldehyde 3-phosphate. Okay, so it's not glucose anymore. It wasn't glucose after the first step. But now we have glyceraldehyde 3-phosphate. We have 2 of those, okay? So there's one on the left and there's one on the right. And so if we continue to follow this down, we can abbreviate that glyceraldehyde 3-phosphate to G3P if we want to, which I do like to, because they're all a mouthful no matter what. Okay, so step 6 we see here is a redox reaction. So what we see, so from here on down, what we're going to see is like this mirror image. So steps 6 through 10, the same thing is happening on both sides of this. It's just duplicated because we took that original glucose molecule and we made it symmetrical, and then we split it in half. So now we have 2 of these G3P molecules that are now going down through steps 6 through 10. So step 6 is a redox reaction. So what we see happening is this NADH plus molecule interacting here and turning into NADH. So NAD plus turns into NADH. So what happens in that process is NAD is gaining an electron and turning into NADH. So that is a redox reaction. So our G3P molecule has been oxidized, so it has lost an electron, and that NADH molecule is what has gained that electron. So we refer often to these NADH molecules as high energy molecules. They're molecules that temporarily store that electron, and eventually that electron is going to get used in the electron transport chain. So for now, it's just being stored in that NADH molecule. And incidentally, that molecule, that NAD that turns into NADH, that molecule is a derivative of a B vitamin. It's a derivative of niacin, and we're going to see that exact same molecule throughout the Krebs cycle as well. And so this is one of those things that, so I think that we've talked about this before, where, you know, when we take in nutrients from the body, there are macronutrients and micronutrients, and our macronutrients are the ones that we can break down to use the energy in those bonds to make ATP. Our micronutrients, we can't break down to make ATP, but they are the machinery that we use, or they're the enzymes that we use in this process. So here we see a B vitamin being used as this temporary storage molecule for energy in this process of making ATP. So you can see that if we were deficient in niacin, this B vitamin, the process would stop right here. We wouldn't, if we didn't have any niacin, we would not be able to get past step 6. That's so then we wouldn't get any ATP out of this process. But thankfully we do have niacin, so it keeps going. So the last few steps, what's happening here is we're continuing to convert that G3P molecule to other molecules, but now we're going to start to get ATP out of it. So you can see steps 7 through 10, we're yielding 2 ATP molecules on each side of this reaction. We're also yielding a water molecule, that's just a byproduct. And so at the very end, the molecule that we have is called pyruvate. And what happens from pyruvate depends on the conditions in the cell. So let's kind of summarize this. Glycolysis starts with 1 glucose molecule. It uses 2 ATP molecules in the beginning. So remember those steps 1 through 3 required 2 ATP molecules. And we get a net yield of 2 NADH molecules. So those are our high energy molecules. It's that B vitamin that's temporarily storing that energy for us, and we're going to see it again later. We have 2 ATP molecules. So 4 ATPs from steps 7 through 10, but then we have to subtract those initial ATPs that we used to get the whole ball rolling. So we have a yield of 2 ATP molecules, and at the end we're left with 2 pyruvic acid molecules. So that's sort of our summary of glycolysis. So what happens next? Well, what happens next depends on whether or not there's oxygen around. If there is oxygen around, then that pyruvic acid is going to go into the Krebs cycle. If there is not oxygen around, then that pyruvic acid is going to convert to lactic acid, and we've heard of lactic acid before. It's a well-known byproduct. Oftentimes we think of muscle soreness as being as having to do with lactic acid, which is not actually the accepted theory anymore, but that's where you may have heard of lactic acid. But it is a byproduct, and it's what happens when glycolysis is all that happens in a cell, where we don't have enough oxygen for the pyruvic acid to continue on into the Krebs cycle. So the Krebs cycle doesn't happen, and all we do is glycolysis. Then we have lactic acid as a byproduct. But that lactic acid is not a loss, because the lactic acid will leave the cell. go into the bloodstream, get picked up by the liver, and the liver will convert it back to pyruvic acid and will use it to make its own ATP. So it will get used eventually, but not by that cell that was producing it. So now let's continue on and take a look at what happens if we do have oxygen around, and we're going to go into the Krebs cycle. So the Krebs cycle, I think I'm going to try and be even more brief about what's happening with the Krebs cycle and let you watch videos to kind of see this process happening. But let's see, Krebs cycle. So there are 9 steps of the Krebs cycle. The Krebs cycle happens in the mitochondria in the presence of oxygen. And again, it is a series of redox reactions where molecules, so we can see here that pyruvate, sorry, it's not super clear, but up on the top we have that pyruvate that we started with from glycolysis. That pyruvate goes through a reaction here where it turns into something called acetyl-CoA. Acetyl-CoA, so we needed this coenzyme A, which is, so underneath pyruvate on the left, you see something where you see acetyl and then you see CoA. So that's acetyl-CoA. That, along with what you see on the left there, that interacts to turn into this molecule called acetyl-CoA. Acetyl-CoA is, well, in order to form acetyl-CoA, again, we need another B vitamin in this process. So here's the next B vitamin that if we're deficient in, the process stops there. So acetyl-CoA, pyruvate turns into acetyl-CoA, acetyl-CoA turns into citrate. So citrate, also known as citric acid, is where the Krebs cycle gets its other name, which is the citric acid cycle, which is what we see on this slide. Then citrate, and so we can see the change in these molecules as we go. Also keep in mind that this is one pyruvate entering here, turning into one acetyl-CoA, but at the end of glycolysis, we had 2 pyruvate molecules. So starting from our original glucose molecule, we're going to run through this Krebs cycle twice, once for each of our pyruvate molecules. So pyruvate turns into acetyl-CoA, acetyl-CoA turns into citrate. This one doesn't show it, but on some models of this, we see each of these molecules as like how many carbons they're made up of, because actually carbons are going to come off of these in this process, and that's where some of that carbon dioxide comes from. That is the byproduct of cellular metabolism. So we see water coming off in that process from citrate to the next step, which is cis-aconitate. Okay, that's that next step. So water is a byproduct there, but then we see water going into the process for the next step. We see another NADH molecule being produced. The next step, there's another NADH molecule being produced, and so on. So this continues around, and essentially what's happening here is, and then we can see if we go all the way up on the left, close to where we started, there's another NADH molecule being produced. So we can see a number of these NADH molecules, and so know that every time we see an NADH molecule, a redox reaction has happened, and that NADH is storing the energy, or storing the carbon. Sorry, not the carbon, I was looking at something else. The electron. The NADH is storing an electron and storing energy temporarily. Also notice that in these, over on the right, each time we have an NADH molecule being produced, there's another byproduct that's being shown there, which is CO2. So that's where that CO2, the carbon dioxide, comes from. That's, and that carbon dioxide is a byproduct, it's a waste product. It will move out of the cell into the bloodstream, and we've seen what happens from there. Eventually we exhale it, but that's where the carbon dioxide comes from that we need to exhale. We have more water being produced, etc. But essentially, let's look at the total, the summary of the Krebs cycle. So throughout the process of the Krebs cycle, including that original conversion of pyruvic acid to acetyl-CoA, which is kind of the sort of an interim step between glycolysis and the Krebs cycle, that process of the pyruvic acid turning to acetyl-CoA. But including that step, we end up with 8 NADH molecules. So again, those are temporarily storing energy. We can't do anything with them but temporarily store that energy there. Eventually we will use them in the electron transport chain. We have 2 FADH2 molecules, which are another high energy molecule that is temporarily storing electrons and storing energy for us. We'll see them come up again in the electron transport chain. We do yield 2 ATP molecules, and as a byproduct we have 6 CO2 molecules. So we saw those where are they all? So there's 1 CO2 molecule up on top in that process of pyruvate turning to acetyl-CoA, and then over on the right where we have those 2 NADH molecules being produced, we have each of those steps has a carbon dioxide molecule. So that's 3 carbon dioxides that we see here. We show 6 carbon dioxides being released total as the byproduct, because remember that we're going to have 2 pyruvate molecules going through Krebs cycle for our original glucose that we started with. Okay, so this is our summary of what we get after the Krebs cycle is 8 NADH molecules, 2 FADH2 molecules, 2 ATP molecules, and 6 CO2 molecules that need to be released from the cell and exhaled from the body. Okay, so our next video, we're going to pick up where we left off here and take a look at the electron transport chain. So we're going to stay inside the mitochondria, continue to see what's happening there, but we're eventually here going to see ATP being produced. So did we do all this? Most certainly we did. We described the role of ATP in cells, so we're totally clear on that. ATP is the molecule that takes chemical energy and converts it to kinetic energy, so it allows for almost every movement that occurs in cells. Any movement that requires energy, ATP is responsible for it. We defined these terms: cellular respiration, aerobic respiration, anaerobic respiration, oxidation, reduction, redox reaction, and glycolysis. We also listed the 3 parts of cellular respiration, which of course were glycolysis, the Krebs cycle, and the electron transport chain. And then we looked at the basic steps of glycolysis, and we talked about where it happens, which is the cytosol, what condition it happens in, which is with no oxygen. So even if there is oxygen around, glycolysis still happens. It just doesn't require any oxygen. And so, but if there is not enough oxygen around for the Krebs cycle to happen, glycolysis can keep on happening in that low oxygen situation and keep on producing ATP for us. But what else will it produce? Lactic acid if there isn't oxygen around. Which is okay because the liver can convert that into ATP. And then we talked about where in the cell the Krebs cycle happens, which is the mitochondria, and the basic reactants. So meaning, what did we start with in the Krebs cycle? We started with pyruvic acid. We converted that to acetyl-CoA and then to citric acid and went on to from there. And then the products of the Krebs cycle, which we looked at, which were all of those NADH molecules and FADH2 molecules and a couple of ATP molecules and a number of carbon dioxide molecules. So that is what we did here, and here is your homework for this video, which is to write out the starting reactant, which is glucose. I'm giving you a hint, and the ending products of glycolysis, which is all given to you on slide 22, and write out the starting reactants, which I guess I have here as acetyl-CoA, but we could start actually with pyruvic acid and then acetyl-CoA and then citric acid, and then the ending products of the Krebs cycle, which is summarized for you on slide 26. So you're just going to write that out for me and email it to me. That's your homework for this for this video, and then next video we are going to get into the electron transport chain and see that process happening in cells. Also on Populi, there is a video for you to watch this process of the Krebs cycle happening, and a video to watch the process of, maybe it's going to be one video, I actually haven't posted it yet, a video with the electron transport chain happening. So we can kind of see that happening in cells, which is sometimes kind of easier to get that visual of these molecules changing and electrons coming off and all of that kind of thing. So so you have those videos to watch, and then the lecture on the electron transport chain and then that's it for this week. And oh, but also posted for you is a Bruce Lipton interview, which is optional but really good. Bruce Lipton has some really good stuff, and I'll tell you about that in class. Okay, so that's it for this video. I will see you in class next week.